Good morning. I hope everybody can hear me. I am Francisco Asturias, Director of the CryoEM core facility on campus. And I would like to start by thanking Vanessa and Dr. Buttrick for organizing this seminar series and giving me the chance to share with you some information about the CryoEM core facility. So the CryoEM core facility is one of the newest facilities on campus. We are located on the first level of the RC1 South building. Access to the facility is through a badge permission. So usually we meet new users in the lobby of the RC1 South building. As I said, I serve as director of the facility, and day-to-day operations are handled by the facility manager, Dr. Eduardo Romero. Information about the facility is available at the facility's website. The link to the facility's website is shown here at the bottom of the screen. So I would like to start by telling you what CryoEM is used for. CryoEM has become in the last 2 years the technique of choice for structural analysis of large macromolecular complexes. And it has been used to analyze all kinds of macromolecules in a wide range of sizes, and all of these complexes perform a number of essential functions in the cell. So in essence, CryoEM can provide three-dimensional structures at near atomic to intermediate and low resolution, and these structures are computed from a relatively small number of images of individual particles. Contrary to what is typical for other structural biology techniques, the larger the complex, typically the better CryoEM works. So in essence, CryoEM then occupies a spot between traditional structural biology techniques like X-ray crystallography and NMR and low-resolution light and electron microscopy approaches. So CryoEM and CryoET basically bridge the gap between these high-resolution techniques and more traditional low-resolution approaches. And CryoEM can cover, as I indicated, specimens that vary in size in several orders of magnitude. The CryoEM facility last year became part of one of the shared resources from the University of Colorado Cancer Center. So we joined X-ray crystallography and NMR spectroscopy to provide a really comprehensive set of tools for macromolecular structure analysis on the School of Medicine's campus. X-ray crystallography is focused on high-resolution, atomic resolution structures of macromolecules. A big bottleneck for X-ray crystallography is that it requires formation of 3D crystals, which basically means generating large amounts of very pure material and running a number of crystallization screens, then solving the phase problem. NMR spectroscopy is great at providing very high resolution information and dynamic information about generally smaller complexes. CryoEM complements these two techniques by really covering a wide range of specimens and being able to generate information about conformation and dynamics of macromolecules starting from very small amounts of material. The facility was established in 2016 with funding from the School of Medicine, and the use of the facility has really grown dramatically over the last 4 years. It first became operational in 2018, and as you can see, we have nearly tripled the number of users in the intervening 3 years. This mirrors what is happening with CryoEM in general across science. What type of services do we offer? So to begin with, we offer consultation services to design and plan CryoEM projects, and we do this mostly through consultations with new users. We also provide data collection and analysis services for multiple institutions in Colorado and neighboring states. And here's just a sampling of some of the places that we have worked with, which include a number of local biotechnology companies. And we also have a robust outreach and training program. We offer individualized training in all aspects of CryoEM to new facility users. I also offer a lecture class on CryoEM every other year as part of the structural biology program on campus. And that class is a combination of lectures and practical hands-on experiences in CryoEM. And we also participate regularly in the Shared Resources Fair organized by the Cancer Center. So what type of equipment is available at the facility? The centerpiece of the facility is a Talos Arctica cryo-electron microscope, shown here on the left. That microscope has a high acceleration voltage and an automated sample handling capability. Is also equipped with a state-of-the-art direct electron detector, a Volta faceplate for looking at dynamic and small particles, and is capable of fully automated data collection and real-time image processing. We also have a second microscope that is used for screening and imaging of specimens preserved in stain, and that is our Talos L120C, shown here in the middle. And in order to prepare samples and keep all of this running, we have a number of sample preparation and ancillary equipment, such as the Vitrobot Mark IV, shown here on the right. The CryoEM facility has dedicated spaces for sample preparation, shown here on the left, as well as a central control room in which all of the computers that drive the microscopes and data analysis are clustered. And the idea of this control room is that it provides an environment where users can interact and share expertise as they use the facility to collect data. So as I mentioned in a couple of slides back, we offer fully automated data collection capabilities driven by an online interface, as shown here. We also have web-based imaging archiving, evaluation, and initial analysis, shown here on the right. This is all driven by very robust IT support from Dave Farrell in the Biochemistry and Molecular Genetics department. And all scheduling and billing for the facility is done through iLabs. We are completing a transition to iLabs that we started last fall. So in order to make all of this happen, the facility counts with a vast amount of computational resources, also funded by the School of Medicine. We have a professionally managed GPU cluster with a large number of GPU cards distributed in 6 GPU workstations that are managed through a queuing system that allows users of the facility to analyze their data. CryoEM generates vast amounts of information. Basically, our scopes generate 2 to 4 terabytes of data per day. Therefore, we have about a petabyte of expandable parallel data storage space that we manage as part of the facility. We also offer professionally updated EM image processing software. Basically, all of the programs that are routinely used by CryoEM users are available at the facility in these workstations. And because we generate so much data, we also have established a system for long-term data storage at the UC Boulder Petal Library. Now, in order to understand what CryoEM is capable of doing, it helps to know a little bit about how CryoEM works. Basically, the way CryoEM works is you take biological macromolecules, shown here in blue. Those macromolecules are preserved typically by freezing them in amorphous ice. This is where the name cryo comes from. The microscope then generates low signal-to-noise images of those macromolecules. The reason the images are low signal to noise is because even after preservation, cryo-EM results in radiation damage to biological specimens. However, those limitations can be overcome through computational analysis. And what is really needed is orientation parameters for each one of these single particle images that the microscope records. And again, by using some sort of math, we can go from this two-dimensional noisy projections of particles to an actual 3D volume of the molecule of interest. Now, something that really makes cryo-EM extremely powerful is the fact that it can take advantage of very sophisticated image clustering algorithms. So if you have, for example, a biological molecule that adopts different conformations, you can use image clustering approaches to separate different conformations of that molecule. And what that allows you to do then is to really gain an understanding of the dynamics of a molecule. And by that, I mean the conformations that the molecule can adopt and how it can interact with other molecules under essentially functional conditions. Now, what type of targets are suitable for cryo-EM? What I have shared with you so far is that it is great for looking at macromolecules in a wide range of molecular sizes, from about 50 to 80 kilodaltons up to several megadaltons. It is also very good at providing structural information about ordered arrays, and these are typical two-dimensional crystals or helical tubes or filaments. Cryo-EM can also be used to look at cells, and that special modality of cryo-EM is known as cryo-electron tomography. What we have on campus is primarily, the capacity to visualize individual macromolecules and ordered arrays. Capabilities for cryo-electron tomography are still limited, but we hope that they will come to campus in the next couple of years. Now, how does a cryo-EM project progress? So before someone is ready to do a cryo-EM, use cryo-EM to do structural analysis of their sample, they have usually gone through a process of isolating and purifying the complex and performing biochemical and functional characterization. At that point, structural information becomes extremely helpful, and it's usually done in two phases. There's an initial characterization done in stain, where molecules are preserved by covering them with a layer of heavy salt. And that allows you to do analysis in 2D and very low resolution 3D analysis, and gives you a sense of the quality of the prep and the rough properties of your complex. Once you have completed this, you then can move to cryo-EM at high resolution. Now, this initial phase of analysis in the stain is typically where cryo-EM projects start prior to the cryo phase, because it really serves as a checkpoint for the likelihood of success for the project. So let me give you an example of what a new user of the cryo-EM facility will typically be faced with. So the person would reach out to us. We would preserve their complex in stain, as shown here. This is an image from one of our microscopes showing several hundred particles of the complex of interest preserved in the stain. Although this seems primitive compared to cryo, it can actually provide a very large amount of potentially very interesting information. It is used to check for homogeneity and purity of the preparation, and it typically provides 2D class averages, such as the ones shown here. Besides doing that, comparison between those class averages can give you already a lot of information about the conformational dynamics and interactions of the complex, as shown here for a specific example with a mammalian transcription regulation complex. So even at this early stage, EM can be used to really provide a surprising amount of information at low resolution about large macromolecular complexes from very limited amounts of material, and that will become clear in one of the next slides. Now, once you have completed the screening and stain, then you typically move to cryo. And here's an example of the capabilities that we have on campus. Here on the right are two structures of a typical CryoEM test specimen, apoferritin, and I am showing here two structures. The one in blue mesh was generated on campus. The one in brown is a state-of-the-art structure at the very limit of what CryoEM can achieve. Our structure is at 2.3 angstrom resolution, so you can very easily distinguish individual amino acids and even fine density corresponding to water molecules. So we have the capability of achieving very high resolution structures of molecules on campus with our equipment using CryoEM. Now, to give you a sense really of how powerful the technique is and of its capabilities, I am using this example from the CryoEM class that I teach every other year. And the students in the class looked at apoferritin, this CryoEM standard molecule, the benchmark molecule, if you wish. And what members of the class did, and these are people that were completely unfamiliar with CryoEM before the class started, and what is shown here from one of the students is a map of apoferritin in which you can easily distinguish secondary structure elements and even see density for bulky side chains. So this map of apoferritin was calculated from data that the students collected and analyzed in class in basically a week or so. The map here specifically came from under 5,000 images of individual apoferritin molecules. So this really gives you an idea of what the technique is capable of. To these images, these 5,000 images of apoferritin molecules came from a single grid. That grid was prepared with 3 microliters of an apoferritin solution at about 20 mg per ml, which means that you can, under ideal conditions, get a 3.6 angstrom resolution map of your molecule from about 60 nanograms of protein. So this really shows you the capabilities of EM, of course, under ideal conditions with a very well-behaved specimen. We have used the CryoEM facility to interact and collaborate with a large number of users on campus. Results from the facility have contributed to a large number of publications and have provided preliminary data for also a large number of grant applications. I just want to highlight here a few of the major accomplishments from use of the facility in the last couple of years. The first example here is from my lab. And this is a large complex. It's called a complex called mediator, which is essential for regulation of eukaryotic transcription. This is a 1.5 megadalton complex with 25 different protein components. And we used equipment in the facility and our computational resources to calculate this map at about 3.5 to 6 angstrom resolution. That map was then used to generate the first atomic model of this huge macromolecular complex. Another example comes from the group of Mamuka Cravastelia in the Division of Infectious Diseases. And this is also work that was done in collaboration with my lab. In this case, we looked at helical tubes formed by the HIV-1 capsid protein in the presence of lenacapavir, a state-of-the-art, new-in-class, potent HIV inhibitor that is completing clinical trials. So information from the CryoEM facility was used to calculate, as I said, these maps of the HIV-1 capsid protein that mimic the structure of HIV capsids in vivo, and to understand the effect of this drug on the structure of these capsids. The punchline from these studies, which were published in Science in 2020, was that the drug stabilizes the capsid, the HIV capsid, and by doing so interferes with progression of the HIV infection cycle. A more recent example from last year came from Jeff Kift's lab. He and Steve Bonilla, a postdoc in his lab, use CryoEM and the resources at the facility to calculate a cryo-EM map of a very small, about 55 kilodalton RNA, viral RNA, that mimics tRNA and uses that exploit to hijack the transcription machinery of host cells. Like I said, this was published in Science last year. And these three examples are just a very small sampling of the amount of information and the number of projects that the facility has contributed during the last 3 years or so. I would like to finish by emphasizing our eagerness to help apply CryoEM in projects that investigators on campus are pursuing. The best way to get in touch with us is by contacting me directly through email or phone. And from that point on, we can devise a plan to work with you and coordinate with Eduardo, the facilities manager, to get your project moving forward. I want to finish by acknowledging and thanking the School of Medicine for funding that made possible establishing the facility, the University of Colorado Cancer Center that subsidizes use for facility users that are members of the Cancer Center and in general provide support for the facility, CERC that provided some funding for some of the equipment, and NIH who has supported all of the research that goes on in the facility. And with that, I would like to end, and I look forward to answering your questions.
Francesca, good morning. This is Natalie. So it's not really like a sophisticated question because I really enjoyed your basics on the cryo-ET. It was really cool, especially taking account how complex it is. But how long is, it's more a practical question. How long does it take for you? And I'm not sure if I'm asking like a single experiment, you know, on your equipment or like a project, but how long it usually takes to, yeah, let's say to complete an experiment, like a single run, as well as a project to get some meaningful data.
Good morning, Natalie. Great to hear from you. Thank you for the great question. That is a very good question. CryoEM is not a high throughput technique. And in order to answer your question, it's good to think about the two ways in which you can apply this type of approach. EM in stain is very straightforward. Basically, it takes about 5 minutes to prepare a specimen. You can get a vast amount. I am talking, you know, many tens of thousands of images of particles preserved in a stain in a matter of an hour. And then overnight, or probably quicker than that, in a few hours, you can generate 2D class averages. So basically, in order to do imaging in stain, you need a vanishing amount of material, and you can get results essentially in a few hours. Now, if you are talking about high resolution analysis by CryoEM, that is a different ballgame. Sample preparation is more involved. Data collection is also more involved. And depending on the quality and amount of the specimen, that could take anywhere from a week to a few months, depending on the amount of data that you have to collect, how well behaved your complex is, how complicated data analysis turns out to be for that sample. So in essence, in the stain, under a day, you can have very helpful information, especially if you're looking at a complex that nobody has any idea about in terms of structure. If you're talking about CryoEM, you're talking more like a week, perhaps with a couple of days of data collection, or up to a few months, depending on the behavior and the complexity of the project.
Okay, yeah, this makes sense. So it means that you actually have to deal with the complex sample prep as well as a very intense computational analysis of the data afterwards. Just as you said, if you're looking for the structure which nobody knows, right?
That's correct. And it really is very specimen dependent. So I gave you the example of apoferritin from the class last fall. Literally, you can collect data in a couple of hours and do live processing while you're collecting data. And the next morning, you can have a structure at 3 angstroms, right? Or overnight, you could have a structure at 3.5 angstroms. If you are talking about something like the complexes that Jeff or Mamuka or I worked with, which are much larger, harder to purify, more dynamic, then you're talking about several days of data collection and very careful screening of the images and convoluted image processing, and then you're talking more weeks and months.
Thank you, Francesca. Thank you.
Thank you for the question.
Francisco, Jim Costello. So kind of along the lines of, Natalie's question in terms of time and effort, how much do you guys consult? So, from us essentially saying we have a molecule, can you guys do everything to helping us with the experimental design? So what kind of services in terms of like consultation do you guys offer?
Yeah, good morning, Jim. Thank you for the question. So for the initial stage analysis and stain, that's, like I said, very straightforward. Typically, it starts with somebody getting in touch with me, and then I coordinate with Eduardo, our manager, to look at your samples. You would then bring, to give you an idea, perhaps 20 microliters of your sample at 100 micrograms per mil, so a very small amount of material. And we can do the imaging and calculate to the averages in a stain for you. Like I said, that will happen in a few days, a couple of days perhaps. In terms of more complex experiments that involve cryo, we can definitely take care of data collection. It's all automated. We have a very robust and stable system to do that. Data processing is more complicated, and it's typically so the bottlenecks for CryoEM, this wonderful technique, are really in two places. One is CryoEM sample preparation. That can be tricky. It's akin to crystallization in crystallography, much less challenging, much more likely to work, but it still, takes some time. And then data analysis can also be complicated depending on how dynamic your complex is. So we are completely capable and routinely help people with projects in stain. Cryo projects, depending on the complexity, typically end up being one of the groups that does CryoEM campus. Did I answer your question?
Yes, absolutely. The other question I had, and this is my naivete with technology, but in terms of like protein interactions, so if we had two proteins that we expected would be interacting, or a protein complex with another single protein, how is CryoEM suited for that, or is something? Yeah, should I be looking somewhere else?
No, that is a great question, and EM would be the perfect technique for that, depending on the size of your complexes and the stability of the interaction. So let me explain a little bit more. CryoEM can detect particles. The lower size ranges about perhaps 50 to 100 kilodaltons. So you saw the example from Jeff Gift's lab of this RNA. RNAs, of course, are very dense in the electron microscope. It would be a little more challenging to visualize a 50 kilodalton protein, but it definitely can be done, especially in stain. So if your two molecules that you're interested in monitoring interaction are over 50 kilodaltons, that can definitely be done by EM. The other issue is the stability of the interaction, right? Yeah. Because in order to obtain images of single molecules, you end up working at very low concentrations typically. So one thing to think about is, what is the KD for the association between these two molecules? Will they remain associated under the conditions that are required for EM imaging? And of course, there are ways around that. So for example, in stain, we end up using very, very low concentrations. In cryo, the concentration of particles that you need could be much higher, up to like perhaps a mg per mil. And there are ways of manipulating sample preparation that will allow you to get around KD limitations. But basically, the bottom line is size and stability of the two interacting partners.
Gotcha. And would fixation, would things like, formaldehyde fixation or kind of binding those molecules together, is that within the scope of CryoEM too?
Yeah, so cross-linking is a very powerful approach for looking at either stabilizing a complex or looking at interactions like you're thinking of. And that typically is done non-specifically with a very, very low, for example, glutaraldehyde cross-linking, or it can also be done with the types of cross-linkers that are used for mass spectroscopy, cross-linking mass spec. So yeah, either one of those approaches could be used to stabilize a complex before looking at it by CryoEM.
Great, thank you. I'll be reaching out to you for more detailed questions that we don't have to discuss here.
Great, looking forward to it.
Natalie, did you want to ask your question?
I feel like a dummy here. So I wrote you, Francisco, I wrote you in the chat. So is it because, I saw both, I saw Cryo-EM as well as I saw Cryo-ET terminology. So microscopy versus tomography. What is like, is it really so different between like 2D and 3D acquisition?
Natalie, great question. I'm glad you brought that up. Microscopy and cryo-electron tomography are basically the same instrumentation, the same approach, right? You preserve a biological molecule in cryo typically, and then you collect images of it. The difference between the two is that in cryo-EM, you have many thousands of copies of the same molecule, right? So like the little soccer player that I used for one of my example slides, you have many, many, many soccer players in different orientations. And you take a picture in the microscope and you may have 200 or 500 individual molecules there, all in different orientations. Then you combine that information computationally to obtain a 3D structure. Cryo-electron tomography, on the other hand, also relies on different views of an object, but instead of collecting images of many identical objects, you collect many images of one single object, for example, a cell. So it's called tomography because instead of taking pictures of 10,000 particles, you take pictures of a cell in different orientations by tilting the specimen in the microscope. You're imaging a single object by taking tilts of that object and collecting images in different orientations. Now, the differences between the two approaches explain their capabilities. So cryo-electron tomography is lower resolution because you have to collect all of your data from a single object, let's say a cell, right? Therefore, you have to contend with the specimen damage. The other way in which they are different is they require slightly different kinds of microscopes. So our best microscope on campus is at 200 kb, Arctica that I showed a picture of. That is perfect for getting to near atomic resolution of single molecules, right? Cells tend to be thicker, and therefore there is a lot more background signal from the microscope. So the ideal conditions for collecting electron tomography data are a higher voltage microscope at 300 kb instead of 200 kb microscope. The higher energy of the electrons reduces interaction with the specimen and limits background noise. And typically a microscope used for electron tomography has an energy filter that discriminates electrons that have inelastic scattering events and contribute to background noise. So we are perfectly capable of doing single particle cryo-EM on campus. We can definitely use our scope for cryo-electron tomography, but it would not be the ideal instrument for that. So the hope is that at some point we will have a high voltage energy filter microscope and we will be able to image larger specimens like cells.
This is perfect. Thanks so much, Francisco.
Thank you, Natalie.
Francisco, this is Mike Metchis. I'm the director of the Animal Behavior and EV Neurophysiology course. I'm sorry if I missed this, but what's your fee structure look like?
What is, sorry, could you repeat
The
your
fee
question?
structure. How do you charge for your services, and what does it generally cost to
Oh, great question. So the facility has a, I think, 3-tier cost structure. We have a price for data collection for users on campus and academic users outside the CU system, and industrial users. In general, access to the microscope is, I believe, around $15 an hour for the small microscope, and perhaps $25 an hour for the large microscope. Those rates go down after the initial few hours of data collection because people typically tend to collect overnight or perhaps over a couple of days. So to give you a very rough sense, I think under ideal conditions, if you had specimens with high particle density and that were well behaved, you perhaps would need about 2 days on the large microscope for data collection. I am thinking that that could add up to about maybe $1,200 to $1,500. And with a good specimen, that could give you hundreds of thousands of images and ideally a near atomic resolution structure. If you work with things that are harder to get at high concentration or that are more dynamic or more challenging, like the complexes that some of the complexes that my lab works with, then you will be talking about larger data collection times, and that will increase the cost of using the facility.
There's an additional charge for the data, the analysis for those images, or?
That is a great question. In general, we offer data analysis services for staying the initial phase to the averages and things like that. We typically do not offer data analysis services for cryo outside the context of a collaboration. And the reason for that is that the facility doesn't have the human resources to do that. So we have computational resources, but doing data analysis for a cryo project can be very time consuming, and it would require, a heavy investment from somebody. So typically the way we do that is either through a collaboration or by providing training and advice and helping somebody do the processing themselves.
Okay, great. Thank you.
Yeah.
Presentation.
Yeah, just to give you a complete answer, there is on the facility's website an hourly charge for data analysis. Is something that we typically tend not to do.
I understand. We have sort of the same thing. The data analysis is so labor intensive that it's sort of a last resort that we do it for. And I think also that when it comes to data analysis, it's often investigator needs to have a better understanding of exactly how their data was produced and analyzed, and it's beneficial for them to do it if they can.
I absolutely agree with you. I think inputs from the person that is interested in the protein, that knows about the biochemistry and function, is essential. So you put it perfectly. It's really two things, right? Lack of resources, and also the fact that the most likely optimal outcome requires the person that knows the biochemistry and function to be hands-on during that process.
Absolutely, yes.
Yeah.
Well, thank you.
Thank you. I am going to check the chat to see if I missed any other questions. It doesn't seem so. So if there are no more questions, I would like to thank you for your attention, and I look forward to hearing from you about potential CryoEM projects.
Thank you, Francisco.
Thank you.
Thank you.
Thank you.
Thank you. Have a good day.